Welcome to Pharmalytics, your go-to source for pharmaceutical science, lab tech, and analytical excellence.
Hello, future innovators and current lab wizards. It's me, Hemu, and boy oh boy, do we have a fantastic journey lined up for you today. We're diving deep into the heart of the pharmaceutical laboratory, a place where precision isn't just a goal, it's a necessity. These aren't just gadgets. They're the gatekeepers of quality, the detectives of drug development, and the unsung heroes in white lab coats, just like this one. In this video, we're counting down the top 10 analytical instruments you'll find in a pharmaceutical lab. Whether you're a bright-eyed student just starting your scientific adventure, a dedicated QC analyst ensuring every batch is perfect, or a pioneering researcher pushing the boundaries of medicine, these tools are absolutely essential for drug development and quality control. We're talking about the instruments that help us understand what things are made of, how pure they are, and how they'll behave. It's like having a set of super senses, allowing us to see the invisible and measure the immeasurable. So grab your safety goggles, figuratively speaking of course, unless you're actually in a lab, then definitely wear them. And prepare to be amazed. We're going to explore these magnificent machines, understand their core functions, and see why they are the bedrock of pharmaceutical innovation. From ensuring the purity of an active ingredient to making sure a tablet dissolves just right, these instruments are critical. Get ready to consider the science because it's all around us, especially in the lab. Let's get started on our countdown.
Kicking off our list at number 10, the UVVIS Spectrophotometer. This fantastic device is used to quantify drug concentration, especially during dissolution, assay, and content uniformity tests. Consider this: light, a wave, and energy. When light passes through a sample, some of it gets absorbed. How much? Well, that depends on what's in the sample and how much of it there is. The UVVIS Spectrophotometer shines ultraviolet or visible light through your sample and measures the light that makes it to the other side. It's like a toll booth for light, and the toll tells us about the molecules within. Think about it. Every drug molecule has its own unique way of interacting with light, its own absorbance fingerprint. By measuring this absorbance at specific wavelengths, we can determine the concentration of a substance with remarkable accuracy. This is incredibly important for ensuring that a medication has the correct amount of active pharmaceutical ingredient, or API. Too little, and the drug might not work. Too much, and it could be harmful. The UVVIS helps us hit that just right Goldilocks zone. It's a cornerstone for tests like content uniformity, making sure every tablet in a batch is consistent. Science rules. Sliding into number 9, it's the FTIR Spectrometer, which stands for Fourier Transform Infrared Spectroscopy. That's a mouthful, but what it does is pure molecular magic. This instrument is primarily used for identifying functional groups in raw materials, APIs, and even polymers. Think of molecules as tiny structures built with different types of bricks. These are the functional groups, like O for alcohols or C=O for ketones. Each of these groups vibrates in a unique way when hit with infrared light. It's like they have their own tiny dance moves. The FTIR spectrometer sends infrared radiation through a sample. Different functional groups absorb specific frequencies of this IR radiation, causing their bonds to stretch, bend, and wiggle. The instrument measures which frequencies are absorbed and to what extent, creating an infrared spectrum. This spectrum is like a unique fingerprint for the molecule, or at least for the collection of functional groups it contains. It's incredibly useful for confirming the identity of a starting material before it even enters the manufacturing process. You wouldn't want to build a house with the wrong kind of bricks, would you? Same principle here. Beyond simple identification, FTIR is crucial for checking the compatibility of different material. It's all about those good vibrations. Molecular vibrations, that is. Drawing up the competition at number 8 is the Karl Fischer Titrator. This isn't about fishing for compliments, it's about fishing for water, specifically determining its exact amount. It's a precise tool for determining moisture content in solid and liquid samples. Now, why is water content so important in pharmaceuticals? Well, too much moisture can affect a drug's stability, its shelf life, how it dissolves, and even its effectiveness. Some drug molecules are like sponges. They just love to soak up water from the air. We call them hygroscopic. The Karl-Fischer titration method is a wonderfully specific chemical reaction. It involves a special reagent, the Karl-Fischer reagent, which reacts quantitatively with water. As you add the reagent to your sample, it reacts with any water present. The endpoint of the titration, when all the water has reacted, can be detected electrochemically. The amount of reagent used tells you exactly how much water was in your sample. It's a very sensitive technique, capable of measuring even trace amounts of water, down to parts per million. This instrument is vital for quality control of raw materials, especially those sensitive APIs, and also for finished products. It's chemistry in action, folks, solving real-world problems. Making a splash at number 7, it's the Dissolution Tester. This apparatus is all about ensuring tablets or capsules release the drug, the active pharmaceutical ingredient or API, at the correct rate and ideally, at the correct site in the body. Think about it. When you swallow a pill, it doesn't just magically work. It has to dissolve first, releasing the medicine so your body can absorb it. The dissolution tester mimics the conditions inside your body, like your stomach or intestines, to see how a drug product breaks down and releases its payload. These testers typically involve a series of vessels filled with a dissolution medium, a liquid that simulates bodily fluids kept at a consistent temperature, usually body temperature. The tablets or capsules are placed in baskets or paddles that stir the medium, and samples are taken at various time points to measure how much drug has dissolved. There are different types of apparatus like rotating baskets, apparatus 1, or paddles, apparatus 2, designed for different types of dosage forms. The rate of dissolution is a critical quality attribute. If a drug dissolves too slowly, it might not provide therapeutic levels in the bloodstream. If it dissolves too quickly, it could lead to a sudden spike in concentration, potentially causing side effects. Checking in at number 6, it's the humble yet mighty pH meter. A simple yet absolutely vital tool for buffer preparation, solution stability, and compatibility studies. Now, what is pH? It's a measure of how acidic or basic a solution is, on a scale typically from 0 to 14. 7 is neutral, like pure water. Below 7 is acidic, and above 7 is basic or alkaline. Why does this matter so much in pharma? Well, many drug molecules are sensitive to pH. Their solubility, stability, and even their ability to be absorbed by the body can change dramatically with pH. The pH meter works using a special electrode that measures the hydrogen ion activity in a solution. More hydrogen ions mean a lower pH, more acidic, and fewer mean a higher pH, more basic. Preparing buffers. Solutions that resist changes in pH is a daily task in any pharmaceutical lab, and you need a reliable pH meter to ensure they are made correctly. These buffers are used in countless analytical procedures and formulations. Holding strong at number 5 is the TOC Analyzer, which stands for Total Organic Carbon Analyzer. This instrument is a superstar when it comes to water quality testing, especially for that ultra-pure water used in clean rooms and for equipment rinse validation. In the pharmaceutical world, water isn't just water. It has to be incredibly pure, especially water used for injections, WFI, or for cleaning manufacturing equipment. Any organic carbon impurities could be a source of contamination, potentially harming patients or affecting product quality. So, how does a TOCI analyzer work its magic? It essentially measures the amount of carbon that's part of organic compounds in a water sample. Typically, it involves oxidizing the organic carbon in the water to form carbon dioxide, CO2. This CO2 is then measured, often using an infrared detector. The amount of CO2 detected is directly proportional to the amount of organic carbon that was originally in the water. It's a very sensitive method, capable of detecting even tiny traces of organic contamination. It's all about keeping things squeaky clean, down to the molecular level. Heating things up at number 4, it's the gas chromatograph, or GC. This instrument is the champion for analyzing volatile substances, things that easily turn into a gas, and for detecting those pesky residual solvents, all as per ICH guidelines. Think about it. Many pharmaceutical manufacturing processes use organic solvents. While these solvents are necessary, they need to be removed from the final product as much as possible, because they can be harmful if ingested or inhaled. The GC works by separating different components in a gaseous mixture. First, your sample is vaporized and injected into a long, thin column. An inert carrier gas like helium or nitrogen pushes the sample through this column. The inside of the column is coated with a special material called the stationary phase. Different compounds in your sample will interact differently with this stationary phase. Some will stick more, some less. This means they travel through the column at different speeds and come out at different times. A detector at the end of the column, like a flame ionization detector, FID, or a mass spectrometer, MS, senses each component as it elutes.
Number 3, HPLC, the Pharma Lab workhorse.
Claiming the bronze medal at number 3, it's the High-Performance Liquid Chromatograph, or HPLC. This instrument is, without a doubt, the workhorse of most pharmaceutical labs. It's used for practically everything: determining the potency of a drug, assay, identifying and quantifying impurities, checking how a drug dissolves, dissolution testing support, and monitoring a drug's stability over time. If there's a non-volatile organic molecule to be analyzed in pharma, chances are an HPLC is involved. Similar in principle to gas chromatography, HPLC separates components in a mixture. But instead of a gas, it uses a liquid mobile phase pumped at high pressure to push the sample through a column packed with a stationary phase. Different compounds in the sample interact differently with the stationary phase and the mobile phase, causing them to separate as they travel through the column. Detectors, commonly UVVIS detectors or mass spectrometers, then identify and quantify these separated components. The high-performance part is no joke. HPLC systems provide excellent resolution, meaning they can separate very similar compounds. They offer high accuracy and repeatability, which are absolutely critical when you're dealing with medicines where even small variations can matter. From research and development, discovering new drug candidates, to quality control ensuring every batch meets specifications, the HPLC is an indispensable tool.
Number 2, the tapped density tester. It's all about the flow.
Nearing the top at number 2, we have the tapped density tester. This might not sound as flashy as some of the others, but it's absolutely key for formulation development and assessing the flow properties of powders. Think about making tablets or filling capsules. You're dealing with powders, lots and lots of powders. How these powders pack together and how they flow is incredibly important for manufacturing a consistent and high-quality product. The tapped density tester does exactly what its name suggests. It measures the tapped density of a powder. You start by pouring a known mass of powder into a graduated cylinder. You measure its initial volume that gives you the bulk density. Then, the instrument mechanically taps the cylinder a specified number of times. This tapping causes the powder particles to settle and pack more closely together. After tapping, you measure the new, smaller volume. This new volume gives you the tapped density.
Number 1, the Analytical Balance precision starts here.
And now the moment you've all been waiting for. Taking the top spot at number 1, the undisputed champion of the pharmaceutical lab, the Analytical Balance. It might seem simple, but every single test, every analysis, every experiment often begins with one crucial step: accurate weighing. These ultra-precise balances ensure your results are reliable from the very beginning. Because if your initial measurement is off, everything that follows will be too. Analytical balances are designed to measure mass with an incredibly high degree of precision and accuracy, often down to 0.0001 grams or even smaller. They usually have a draft shield, a clear enclosure around the weighing pan, because even a tiny breeze or a change in temperature can affect the reading of such a sensitive instrument. Think about that. They are so sensitive your breath could change the measurement. That's why they are often placed on special anti-vibration tables in temperature-controlled rooms. Whether you're weighing a tiny amount of a potent API, preparing a standard solution, or determining the mass of a sample for any of the other instruments we've discussed today, the Analytical Balance is your starting point. Calibration is paramount. These balances must be regularly calibrated with certified weights to ensure they are performing correctly. It's the foundation upon which all other analytical data is built. Without accurate weighing, the sophisticated science performed by all those other amazing instruments wouldn't mean nearly as much.
And there you have it, folks. Our countdown of the top 10 must-have analytical instruments in a pharmaceutical lab. From the foundational precision of the Analytical Balance to the molecular insights of spectroscopy and chromatography, these instruments are truly the foundation of quality, safety, and innovation in the pharmaceutical world. Each one plays a critical role, a specific part in the grand orchestra of drug development and manufacturing. Mastering their use, understanding their principles, and appreciating their capabilities truly means mastering your lab career and contributing to better health for everyone. Science is all about tools, and these are some of the best. These instruments, working in concert, allow scientists to develop life-saving medicines, ensure their purity and potency, and make sure they perform as expected. It's a world of meticulous measurement, careful calibration, and constant curiosity. The next time you see a medication, take a moment to consider the incredible science and the sophisticated technology, like the instruments we've explored today, that went into making it safe and effective. It's a powerful reminder of how science impacts our daily lives in the most profound ways. Which one of these incredible instruments do you use most in your lab? Or which one do you find the most fascinating? Let us know in the comments below. We love to hear from our community of science enthusiasts. And hey, if you found this journey through the Pharma Lab as exciting as I did, don't forget to like this video, share it with your fellow science explorers, and subscribe to Pharmalytics for more lab insights and analytical adventures. Keep asking questions, keep experimenting, and keep considering the science.
Thanks for watching Pharmalytics, where science meets precision.